Hey guys, welcome back from Thanksgiving break. We're going to start off this with mitosis. Now, for most of us, I'm hoping this is a review. I think most of us did mitosis in 7th grade life science. We're going to go in a little bit more detail and kind of a big review. Okay, so let's get to it. All right, so in eukaryotic cells, remember eukaryotic cells, something that has a nucleus and membrane-bound organelles. The genetic information is passed from one generation to the next. This is done via chromosomes. Okay, we'll get into more DNA and RNA and all that kind of stuff in a little bit. Chromosomes make up. All right, so the cell cycle is the cell cycle. So it is the entirety of all that's going to go on with the cell. So if I'm drawing it up here, all right, gotta look at it like a clock. And this is the example you're going to see a lot. And it's going to show you like certain sections. So there's like this section, there's like this section, like this section, a tiny section right here. All right, so this cell cycle takes into account everything that the cell is going to do. Its growth as it prepares to split, when it actually divides and we form the two new daughter cells. All right, so the cell cycle is everything, not just mitosis. There are 4 phases: G1, growth 1, synthesis, growth 2, and M phase. And M phase also kind of takes into account cytokinesis, which we'll talk about. And here we go. Okay, so in this first phase we got G1, which is where we're going to do normal cell things. Okay, S, we synthesize our DNA, we prep up for M phase, prophase, metaphase, anaphase, telophase, or PMAT, and then we have cytokinesis at the very end. All right, so cell cycle. We got interphase. Interphase takes into account all the first 3 phases of the cell cycle. So G1, S, and G2. So here's the whole entirety of the cell cycle. The numbers are a little off, or the measurements are a little tiny bit off, but you see right here, this 3/4 of this is interphase. Probably even more than that is interphase of the life cycle of the cell. So you got G1, S, G2, and then short little mitotic phase, which is where it's actually going to do division. But most of the time it's sitting like doing cell things, then it's getting prepped up for division, and then it divides. So G1 is where the cells spend most of their time growing, increasing in size, synthesizing new proteins. Essentially, they're doing whatever cells do. So if it's like a liver cell, it's doing liver cell things, skin cell doing skin cell things. Whatever depends on what it is going to be what depicts what it does. Okay, S phase. So here's where actually, like it says, synthesis. It's kind of what the S stands for. Chromosomes are going to start to replicate. We'll go more in depth in that in a little bit. We're going to make or synthesize DNA. All right. This is the shortest of all these phases of the of interphase. Organelles, they're all getting prepped up for division. So key here is division. DNA is replicated, yep, in the last phase, but everything is getting ready for division. Organelles are getting ready for division. The molecules, just general stuff that is inside the cell, all of it is getting ready for division. All right, now we actually get into M phase. So for M phase, this is only in somatic or body cells. And we're going to divide these up in just a minute, or a little bit, because we'll talk about meiosis. But somatic cells are body cells, meaning non-sex cells. Okay, sex cells, sperm and egg for like us and mammals and anything that reproduces sexually is going to use sex cells. So that'd be gametes. So somatic cells are just regular old body cells, skin, liver, lung, tissue, brain, blah, 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 blah. List can go on forever. You don't have to write this because we're going to talk about each more in depth, but it consists of prophase, metaphase, anaphase, and telophase. And then cytokinesis is this last little part, kind of part of telophase, kind of not part of telophase, where we're just going to split the cytoplasm. All right, so prophase takes the longest. All right, chromosomes become short, fat. Chromosomes turn to chromatid. All right, make sure you know these. All right, chromosomes to chromatid. All right, and then sister chromatids join at the centromere. So we've all seen that classic kind of X shape of chromosomes. All right, so here's one sister chromatid on this side, the second on this side, and they're joined in the middle. All right, so draw some little arrows, which I'm sure I have a picture coming up here in a minute, but I like to draw. Ooh, okay, there's your centromere. And the nucleolus, remember that nucleolus is inside the nucleus, starts to disappear. All right, centrioles, we talked about these last time. They are helping in cellular division. They're moving to opposite ends. All right, north, south, east, and west, however you like to look at it. Spindle fibers start to project from the centrioles, and they're going to grab the centromeres. All right, so chromosomes in the form of chromatid attached at the centrioles by spindle fibers. So you'll see right here, you got your centrioles. They shot out the spindle fibers. They're going to latch on to the centromere. And eventually, we're going to be moving into the next phase right here. Okay, nice little visual representation. So nucleus is almost gone. Nucleolus and all this is almost gone all around here. You can see it breaking up. Centrioles would be right here. They look like little churros. Okay, shooting out the spindle fibers here. We're good to go. There's a cell in late, or probably mid prophase, yeah. All right, next, metaphase. Meta-middle. Okay, so what we're going to do is our chromatids are going to arrange along the equator or the midpoint of the cell. And that's really about it for this one. So what's going to happen is centrioles shot out the spindle fibers. It kind of grabbed a hold of the X. Let's say, you know, right here, one's X right there, one X right there, one X right there. Okay, and it's going to take it and it's going to push it to the middle. Okay, so you'll see that each centriole has shot out its individual spindle fibers, and they're going to kind of latch on here, right, and prep for the next part. So you can see they're all kind of lined up on the middle right here. This is in a plant cell, but you can tell by the cell wall. Anaphase, they're going to be pulled apart in anaphase. So at the centromere that joins the sister chromatid, they're going to separate. So they're just going bloop. One goes one way, one goes the other. Now the sister chromatids are identical. So what they're doing is they're splitting apart. That way equal sets of chromosomes go in each side. So it can be like, all right, we got 46 chromosomes, like in our cells, and the next cell is going to have like 42 or 44 or 48 or 49. All right, this is no bueno. So we have to have 46 in each one, 23 and you know, for mom, 23 from dad, makes you 46. All your body cells, or the somatic cells, are going to have 46. So when they line up on the middle, they have to get separated out evenly. So this is, you know, kind of the cell's way of making sure that we get everything separated evenly. So they move to opposite poles, like I said, because the poles are going to become, they pull apart, and then eventually we're going to pinch off that little section in the middle and make two new cells. So you can see we were lined up in the meta, we were lined up along the middle, get pulled that way, the spindle fibers shorten, everything gets pulled back, and we end up with, eventually, we'll end up with equal numbers. So this bad boy, it had 8 before the division, now has 4 in each one. So it started with 4, chromosome number doubled to 8, then we went back to 4 in each cell. So like G1, then we got by the end of S, all right, and then when it's all done, when mitosis is done, we'll be back to 4. All right, telophase or telophase, you'll see them both written, said the same way, or used interchangeably. Chromosomes become long, skinny, not so clear. So what they're going to do is that soupy stuff in the beginning and S1 that became like strands of spaghetti and then ultimately condensed into chromosomes and then the chromatid, it's going to like redo or undo that process. So it's going to slowly start unwinding, start becoming more of this soupy mess as opposed to clear defined chromosomes. Spindle fibers break up, and then as they pull apart and as they get more and more apart, right, we're going to kick in and we're going to do the cytokinesis, which cytokinesis just really means the splitting of the cytoplasm. So here you can see in telophase you're going to have this. Eventually right here is just going to keep pulling apart and bloop, plop into 2 new cells. So once again, what is cytokinesis? It's kind of occurring at the same time. It is and is not. It's kind of own little part. It occurs at the same time as cytokinesis. And it's just where everything's pulling inward. And then eventually it's going to pull apart. Once it becomes pull apart, the cells are going to start cell cycle over again, and they're going to start back up at G1, growing, doing their things. All right. But cytokinesis, all we're doing is just pinching the rest of that cytoplasm apart into 2 new cells. Now, the difference between cytoplasm or cytokinesis in plant cells and in animal cells is plant cells, pull apart, become 2 new things. But in plant cells, so here's my cell. All right, here I got all my chromosomes over here now. All my chromosomes over here now. Well, due to the cell wall, they can't really pinch apart. So what happens is slowly, back and forth, back and forth, back and forth, it'll start to lay down cellulose, or that material that makes up the cell wall, until over time, you have I can't draw a straight line to save my life. You will have a cell plate that cuts it in 2. Okay, so this cell plate is what's going to cut this plant cell and kind of finish cellular division in plant cells. So you can see kind of here you got, really in this one, you can see this cell plate forming right there. Here we have an animal cell where you can see it kind of pinching in and around, kind of this round shape right there. All right, so a few things. Last but not least, protein or cells have things that help regulate this. These proteins are called cyclins. They kind of trigger to turn on and off cellular division. All right, so skin cells are going to be doing cellular division at a much higher rate than like liver cells or lung cells or so forth and so on. So each cell kind of carries their own cyclins, which help it regulate how fast it's going to divide. Cancer is when these cyclins that normally would tell a cell to stop division or go on division are just, they're not working right, and they become too much and they continue to grow and grow and grow and grow. Of course, for better or worse, I'd say for worse, We've all of our lives have probably been touched by somebody with cancer, and you can either have malignant, which is the not so good kind, or benign, which is kind of they would usually go in and cut out, and the person would hopefully make a full recovery. All right, guys, that's it. Please make sure you take the notes. Come in with questions tomorrow.